All right, well, first of all, let's talk just a minute about the exam. All right, I think that's this. Well, I can't go over any exam questions yet because we have a few sick people that are yet to take the exam. I can share with you the distribution. We're getting a lot of A's, which is good. A little bit, you know, higher on the bottom end, obviously, than I hope to see. With the curve, the mean was 75. So I did curve it up actually quite a bit. I did it a little creatively again this time, where instead of missing 1 point, I only took off 0.8 instead of 1. So I multiplied 0.8 by the number wrong. And then I added 4% to everybody's score as well. So again, if you want to kind of work out what that meant for your score, like before and after, I'm not sure if that's really terribly relevant, but if you're curious, you're welcome to do that. Again, I would encourage you all to come and look at your exams in my office. If my office hours don't work for you, then we'll find another time that works. Okay, so I think that is always a good thing to do. I am very careful about errors. I have kind of a system that I go through, not to say that I don't ever make mistakes, but once I go through an exam, I take a look at the people that only missed a few, and then look at the ones they missed and make sure that that I didn't key out any errors there. But, okay, so just, you know, for transparency, that was the distribution of the class. Okay, again, we will go through some specific questions once we get everybody to take the exam. All right, so we're kind of switching gears a little bit. Up to this point in class, we've been really talking about the basics of different kinds of microbes and how those cells work, their metabolism, how they reproduce, how we grow them. We're really switching gears now into some application. So we're thinking now for a while, how do we kill microorganisms? In particular, how do we kill bacteria? So this lecture, we're talking about, you know, how do you get things to be bacteria free? How do you get rid of microbes in our environment? Next week, we're going to start talking about antibiotics, how they work, some specific antibiotics, how they work. We'll talk through kind of the process of infection after that, and then we'll get through how does our body's immune system work to try to kill microbes in us. So again, thinking a little bit more application. So chapter 9 here is control of microbes. You know, historically speaking, before about 1860 or 70, we didn't really know what caused infectious disease. So if we were going to try to prevent ourselves from getting sick, really it was just all about superstition. Okay, so some things that people thought maybe might help control disease, bathing in urine a couple times a day. They thought that might be helpful. Wearing a dead toad around your chest. I couldn't find a picture of that one. Tying a live chicken to the site of infection. I can't even like imagine doing that. I hate chickens. I had a bad experience with a rooster once. Like, whoa, I can't even imagine that. The ever popular leeches, right? If they thought, you know, bad blood caused disease, then get rid of the bad blood. That was popular for a long time. Back in the Middle Ages, when bubonic plague was a huge problem, that was personal protective equipment in the Middle Ages for doctors. They would wear long robes, they would try to cover up all their skin, and they wore these beaks that had herbs in them, and that was supposed to kind of counteract the bad air that they thought maybe infection was caused by bad air. All right, so we've come a long way. All right, so what are we going to talk about in this chapter? We have to kind of go through some definitions first. We're going to try to use words correctly. And then we're going to think about kind of just the parameters that we have to consider when we're choosing what method to use to try to get rid of microbes, whether it's on a surface or whether it's on our skin. And some of the two of the physical ways that we're going to kind of talk about the most is the use of heat and radiation. Just explore different ways we use those two things. And then we're going to talk about some different chemicals that are used as well. I'm not going to talk about as many that are in the book, so it just kind of makes things a little bit more manageable. And then I hopefully that you'll learn some things even about maybe cleaners that you've used for a long time, like why is bleach come in a white container, or we're not gonna go through how to wash your hands, but what are the advantages of hand washing versus using hand sanitizer? So again, hopefully things will be a little bit more familiar because they're terms that you've maybe dealt with. Although you might not have paid attention to them your whole life, you should be able to draw, connections to your life, because, right now everybody's thinking about how can we stay germ free. So very applicable topic. All right, so I used to kind of write out a lot of these terms, and then I realized, oh, there's a nice table in the book that really kind of sums everything up really nicely. So some of these terms that we'll be using this chapter, and this really dovetails really nicely with what you've been doing in lab this week, if you're in lab, so it's nice to have a little crossover. These terms, antisepsis and disinfection, or an antiseptic versus a disinfectant, right? These are both chemicals that we use to reduce microbial numbers on surfaces, but by definition, something that's an antiseptic is going to be used on living tissue. And then something that's a disinfectant will be used on non-living tissue. All right, so usually antiseptics are going to be a little bit less irritating, less caustic, oftentimes less concentrated. And then when we, concentrate, say, the chemical, for instance, then it's going to work a little bit better, but not necessarily be safe to use on our skin, for example. An aseptic procedure, if you're in lab, you should recognize that. We talked about that in here as well. These are just things that we try to do to reduce microbial numbers. So things like flaming your loop. Let's see, these suffixes, side and sidal, and we can compare them to the suffixes stasis or static. If a chemical is bactericidal, that means it's going to kill, destruction of that microbe. So bactericidal destroys bacteria. Virucidal destroys viruses. Some chemicals or methods of microbial control may not necessarily kill the microbe, they just may inhibit it. So something that's static, like a bacterial static measure, will just keep bacteria from growing in numbers. Won't necessarily kill it outright. And sometimes that's good enough. Something that's considered a degerming technique, that just usually refers to some kind of mechanical scrubbing. Okay, so that's kind of part of why washing your hands is effective, because you're scrubbing those microbes off. Let's see. And then these three terms, pasteurization, well, actually, let's talk about that. Pasteurization, we've talked about that briefly already. We talked about how Louis Pasteur first controlled the spoilage of wine by heating up grape juice a little bit to get rid of the bacteria in it. All right, so we're going to talk much more about pasteurization, commonly used to reduce microbial numbers in milk and other liquids like apple juice. And then the difference between a sanitation and a sterilization. A sanitation is going to reduce microbial numbers. Usually we talk about something being sanitized to meet some public health standards. So if you've ever worked in a restaurant or even like a concession stand, you can't just wash the dishes. You have to have an extra sanitizing step where you're dipping the dishes into a bleach solution. Okay, so usually, again, in restaurants, we might have kind of a little bit more stringent requirements that need to be met more so than what you would just do at home. And a sterilization is total destruction of all microorganisms, even spores. All right, so that level is pretty difficult to attain. There are some chemicals that are considered high-level antimicrobials that will destroy everything. But most often you need heat in order to totally kind of destroy all the microbes. So I will try to use these terms correctly. Again, just so, you know, they're not always used correctly in everyday language. Okay, so thinking about, before we get into like our specific methods of how to kill microbes, thinking about why they do. Sometimes we don't really know, okay? The microbe dies, all right? Might be a combination of things that a certain maybe chemical acts on. But sometimes we can be kind of clear about that. And there's two really basic mechanisms that we could use to describe how microbes die. Either their cell walls and membranes are altered. Okay. So if their cell walls and cell membranes are not intact, think about the ways that that's going to affect the microbe. If the cell wall is damaged, then we know we're kind of jumping ahead. This is how a lot of antibiotics work. But if the cell wall is damaged, then they're a little bit more fragile in terms of differences in osmotic pressure. So water can rush in and the cell can burst if their cell wall is not intact. The loss of membrane, its integrity, can cause all sorts of problems. It can cause things to leave the cell that shouldn't normally leave. It messes up that proton motive force. So the protons are leaking in places other than that ATPase enzyme. So it affects the cells, a bacterial cell's ability to make ATP, certainly. And if we're talking about a virus, if an envelope virus loses its membrane or it's kind of not intact, then that virus probably is not going to be able to attach to its host because the proteins for attachment are going to be in whatever the outside layer is of the virion. So if it's membraned, then it loses ability to attach. The other basic method, the way that antimicrobial agents work, is causing damage to proteins and DNA. Okay, so we've talked a little bit about mutation already and some kinds of chemicals that can cause mutation. Obviously, if the 3D shape of a protein is compromised, then usually that protein is not going to be able to function. So bonds are broken by whatever treatment. Any kind of mutation is going to alter the nucleic acids, and then the organism can't reproduce correctly. So, okay, so again, sometimes we don't really know exactly what it is. It might be a chemical does all those things. Okay, so I'm not making you write a lot today, but this one slide I am. So if we think about kind of what antimicrobial to choose, you've got something that you want to get clean, and well, what do? You use a chemical or something else? Well, there's a lot of factors that you have to think about. So thinking specifically about the site to be treated. Okay, is this a living object, sorry, a living, part of a living organism that you're trying to cut down microbial numbers, or is it some kind of surface? Is it okay to leave residue on that surface? Is it okay to stain it? Okay, those are things that you have to think about. Exposure time. Most of our antimicrobial methods require at least some exposure time. And it's easy to forget about that. I know at home, even I'm guilty of this too. I spray, bleach on the counter and then I wipe it off. If you look at the container, it usually says, we'll kill 99.9% of microbes with 30 seconds of exposure time. So you're supposed to spray it on and leave it there a little bit and then wipe it off. Again, keep in mind, how much time do you have? Does this have to be something that kills on contact, or can you afford to give it some exposure time? The concentration of, say, this is a chemical means, may be really important. Usually if it's less concentrated, it's less effective. Not all the time. We'll talk about the exception there. But if you can dilute it, then it's going to be cheaper. So kind of depends on what you're trying to kill there. Are the cells that you're trying to kill, are they actively growing? Cells that are actively growing are a lot more susceptible to being killed. It's easier to kill them if they're actually growing. What about the temperature? Okay, we know that for a lot of methods, cells are going to die quicker when the temperature is higher. So for this particular treatment, I think it was, I don't know, this is a figure from your book here. I don't know. I think it was, they were being treated with some chemical. The cells at 45 degrees Celsius, which that's pretty hot. That's above 100 degrees Fahrenheit. Only took about 4 minutes to kill them all. But at room temperature, the killing time was a lot slower. It took more like 10 minutes to kill the cells. So in higher temperatures, metabolisms are faster for cells, and they're probably going to die quicker. What about the pH? Some chemicals are going to work better at certain pHs. So if you've ever taken care of a pool, then you know that you have to pH the pool water very carefully, because the chlorine will start to dissociate as the pH gets higher, and then it won't be effective anymore. So you want to keep the pH in the range that it's going to actually, the chlorine's actually going to work. Okay, and one last one here that's easy to forget about is organic matter present. Okay. So what I mean by organic matter, dirt maybe, but also things like blood, feces, diarrhea. It's tempting when you've got a big mess to clean up, like your kid throws up, throw the bleach on, wipe it off, be done with it. But the bleach is not going to penetrate very well through organic matter. I know that a few years back, I was on a mission trip, in a third world country, and lunchtime came and we'd all whip out our hand sanitizers and think that we were good to go. Well, that hand sanitizer, that alcohol doesn't penetrate very well if your hands are dirty. So, it was the best we could do, but it wasn't working as well as it could have, that alcohol, because, our hands were certainly dirty. Okay, so you have to usually clean up a mess and then disinfectant, then add your bleach or your chemical or whatever, because a lot of times, it depends, again, on your method, but it might not penetrate through that very well. Okay. It's also important to think about what kind of organisms you are trying to kill. All right, so when I look at this list of microbes, and when you look at it, hopefully you think, wow, I know what all that stuff is now. Like I have learned a lot so far this semester. I don't think that you need to memorize this list, like hardest to kill, easiest to kill, but you should kind of have an idea of the really hard ones and the really easy ones to kill. So this is kind of relative susceptibilities. So there's going to maybe be some wiggle room here when you start talking about specific genera of organisms. And of course, we're talking about like all antimicrobial agents. So it depends what specifically you're talking about, whether it's a chemical or whether you're talking about radiation or whatever. But in general, you know, prions are definitely the most difficult things to inactivate. If you are going to use heat to inactivate a prion, you have to use 900 or 400 degrees Celsius heat, which is 900 degrees Fahrenheit for 4 hours. All right, so it takes a lot of heat to deactivate that. We do have some chemicals that will deactivate, you know, instruments, like medical instruments, but you have to soak the instrument in there for like hours and hours, 10 hours of exposure to deactivate the prion. So thankfully, those are pretty rare and we don't have to deal with those very often because they are very hard to kill. More commonly, we might come in contact with bacterial endospores. Those are difficult to kill. Less so the cysts of protozoa. It depends what kind of cysts they are, but that's another kind of resting stage. Again, not quite as tough as endospores. The mycobacteria we talked about, those are the acid-fast cell walls, have that mycolic acid in their cell wall that makes them pretty impermeable to a lot of things. So that's kind of the top of the list. Easiest thing to kill, envelope viruses, right? So that's COVID, it's an envelope virus. So luckily, that's pretty easy to kill. I think I mentioned that a couple lectures ago, that those membranes just, they dry out really quickly. So we're talking about in the environment now, they degrade very quickly. This talks about, you know, gram positives as being easier to kill than gram negatives. I think that that peptidoglycan just is kind of really permeable to things, but I don't have really a good explanation why that would be the case. So again, you know, don't worry about kind of the mid-range things here. That's just kind of a general idea. Okay, so if you're only needing to kill things down at the bottom of the list, you might choose a different chemical than if you really need to kill like everything up at the top. All right, so kind of looking at, these are the methods that we're going to look at in a little bit more detail. We're going to start with some physical methods and then get into chemical agents of control. I have not chosen to talk about all the ones that are in your book. Okay, so there will be some things in your book, especially some chemicals that we're not going to talk about. We're going to spend a little bit more time on the heat and the radiation. And, you know, this is really pretty straightforward stuff. So we're going to kind of blow through the rest of this pretty quickly. And I'm not going to make you do very much writing. Okay, and again, the chemical agents I've kind of organized by low, intermediate, and high levels, and just kind of giving you a few examples here. So I'll try to give you an idea of, you know, where I'm kind of throwing a lot of information at you, but try to give you kind of an idea of where to focus your studying. Okay, so heat. Again, if you are in lab, this is what you talked about in lab yesterday and today. How does heat kill microbes? Basically, it does all sorts of damage. So denatures proteins, destroys membranes, cell walls, disrupts the structure of nucleic acid. It does everything. It's very thorough. Okay, so what we were testing in lab this week is we're trying to measure a way to measure an organism's tolerance to heat. So some organisms can just withstand higher temperatures than others. Okay, so one of the things that's helpful to test, is the thermal death point, sorry, thermal death time. Okay, so if your lab this week, you exposed some cells to a water bath at elevated temperature, and then took out aliquots, at different times on a plate, then we're going to incubate that plate and see how long that organism was surviving. So hopefully next week you might get some data in your lab that looks something like this. And I just made this up as to whether that organism grows times 0. That was just our positive control before we exposed the organism to heat. And then, took out aliquots at different times. So for E. coli, what would be the thermal death time, the shortest time that killed? Okay, that's going to be 4 minutes. Okay. Now, you could do the experiment again and test 2 minutes and 10 seconds and 2 minutes and 20 seconds. Like, we could refine that further. But, for our purposes, 4 minutes. It's dead at 4 minutes. Staph aureus would be the same thing, the shortest time that kills there, so 4 minutes. Usually an organism like Bacillus is going to be living a lot longer because it's a spore former. All right, so if there were spores in that medium already, once you get it in the nice, nice temperature of the incubator, those spores will germinate when even all the other vegetative cells would have been killed by the heat. Okay, the other thing that you can measure, which you're kind of switching the variables a little bit, is the thermal death point. Okay, that's the lowest temperature that kills at a given time. All right, so to do this, we have to put our organism in, we would have to test at different temperatures of water baths usually, so it's a little bit more cumbersome to do. But again, I just made up some data here. For E. coli, the lowest temperature that kills here is going to be 60. For Staph aureus, 65. And then for Bacillus, the best we could say would be greater than 70. Again, those spores are going to withstand pretty high temperatures, at least for short periods of time. All right, so we know that there are differences in organisms and what they can withstand. So if we think about some applications of using moist heat, generally using moist heat is going to be better than dry heat because it penetrates more quickly. But again, it depends what you're trying to get rid of the microbes on. If it's something that's, was going to be ruined by getting wet, then obviously that's not going to be a good thing. And again, those of you in my lab kind of heard the same rundown today or yesterday already. Putting something in boiling water obviously is going to cut down on the numbers of microbes on it. You could either consider it though a disinfection or a sanitation. If you're just at home, it would be a disinfection. Probably, like I said, public health setting, then you would more call it a sanitation. Not a sterilization because endospores actually withstand boiling water for about 30 minutes. So it's enough to kill everything else within about 10 minutes, but because it takes so long to kill spores, it's not considered a sterilization. Pasteurization. It's a topic that, I don't know, for some reason interests me a lot. So maybe I'll tell you more than you want to know about pasteurization. But we pasteurize our milk now, not only to renew, well, when we reduce microbial numbers, then it also increases the shelf life. Okay, so, obviously we go to the store, we buy our milk in a refrigerator, and it's usually good for a couple weeks. There's a date on it. Here's a hint, it's going to be good way past that date on your milk. So if you're one of those people that it's the day after and you throw it out, it's safe to drink actually way after that. We have tested this in another lab that I've been. Even when it starts to get chunky, it's going to taste nasty, but it probably won't make you sick. Okay, so just kind of something to store back in the back of your head. In this country, we typically do this flash pasteurization, where we bring the milk up to about 72 degrees Celsius and leave it that way for only 15 seconds. And so we're not boiling the milk, so it's not killing everything, but it's killing all the organisms that are likely to make us sick. So things like Mycobacterium tuberculosis, TB, Listeria, E. coli. There's all sorts of microbes that grow really quickly in milk because it's a really rich medium. Sterile in the cow, as long as the cow's healthy, but then as soon as it comes out, it gets contaminated really quickly. So we've been pasteurizing in this country since like the 20s, the 30s. Michigan was the first state that said that milk had to be pasteurized in order to be sold, and that wasn't until the 1940s. But again, before that, lots of diseases were passed in milk. Today in this country, our cows are very healthy and they're milked very hygienically. So the CDC says that it is safe for people to drink milk that hasn't been pasteurized. So we call that raw milk. Unless you're pregnant or immunocompromised, and then you should only drink pasteurized milk. But in other countries, like over in Europe, they do these ultra high temperature pasteurization or ultra high temperature sterilizations, where you're bringing that temperature up really high, really briefly, but that's enough to sterilize the milk. So then it can be packaged in containers that you don't have to refrigerate. So you buy milk at room temperature, and it keeps for a really, really long time because there's essentially no microbes in it. It changes the flavor a little bit though. So that hasn't really caught on in this country. Okay, so again, in this country, this is a sanitation, just reducing microbial numbers. Okay, and then if we really, really want to get really serious about getting rid of all the microbes on some object, we're going to autoclave it. All right. So, I found this picture just online. That's the way most autoclaves look. They look pretty nasty and dirty. They're these big, metal contraptions. They are going to use steam under pressure in order to sterilize. So kills everything. Okay, let's like that to sterilize. Okay, so to make sure that everything's sterile, you set the autoclave to come up to 121 degrees Celsius. So how do you get water to 121 degrees Celsius? Usually when water boils, it turns to steam. Well, that's why we pressurize it. So when you increase the pressure, the boiling point actually increases. So we can kind of superheat the water at 15 pounds per square inch. That's an additional atmosphere of pressure that we put it under. And then you only have to keep it like that for about 15 minutes. And everything in the autoclave then is going to be sterile. Okay, so how do you be sure that your autoclave is working correctly? Ours upstairs breaks down regularly. So we always put autoclave tape on it. So say it's some media that we're trying to sterilize. The tape looks like this when we put it on there, and then after it comes through the autoclave, those bands should turn black. And that's a fairly reliable way to know that, yes, it's come up to the correct parameters and whatever's in it should be sterile. This is just kind of the inner workings of an autoclave. You don't have to understand that, just sort of if you're interested, but again, it's kind of, complex again, because we're bringing up the pressure there. Okay, so some things, you can't get wet, but we use an autoclave for, glassware and swabs and just about everything in microlab. If you're trying to sterilize something like an oil or a powder, then it's not going to be the best. We're going to have to find some other way to sterilize. Okay, so we could use, instead of moist heat, there are some applications for using dry heat, again, for something like oil that you can't get in contact with water. A hot air oven It can be used to sterilize as well. It takes a longer time and it takes higher temperatures. But again, you can use it for things that can't get wet. Usually you'd bring the temperature up to 160 degrees Celsius, which is only like, I think it's like 320 or 340 degrees Fahrenheit. So it's not super hot, but you got to leave it like that for a couple hours in order to have the same sterilizing power as what an autoclave would. Okay, and one very useful application of dry heat is incineration or burning. Okay, we use that all the time to sterilize our loops in microlab. Waste disposal, we'll just burn it, our biohazardous things, and then we can be sure that it's not going to get anybody else sick. All right, so other ways that we're going to use to limit microbial growth. So hot temperatures work. What about cold temperatures? Well, cold temperatures are only going to slow bacterial growth. They're not typically going to kill cells, or, you know, if they kill some of them, if they're not likely to make kind of the whole culture viable. Refrigeration, again, slowing bacterial growth. So we might say it's bacteriostatic. Okay, static means it's slowing growth. So we do this all the time in lab. When you grow up your cultures, we incubate them overnight, and then we put them in the refrigerator till you all come back to lab again. Otherwise, your cultures would overgrow. So we slow them down, make sure they don't grow anymore. Now, there are exceptions to that. There are organisms, remember, psychrophiles are ones that grow optimally in cold temperatures. And there are organisms that are considered facultative psychrophiles. So like Listeria, that's an organism that grows optimally more like room temperature, but it grows really well in the refrigerator as well, not quite as fast. And that's a common foodborne pathogen. Every once in a while you hear about maybe some lunch meat or something being recalled due to finding some Listeria contamination, you know, at the processing plant. Freezing, you know, same thing. It just really stops growth, doesn't usually kill cells outright. Oftentimes, if we have some cells that we want to store for long periods of time, we'll actually put them at minus 80 degrees Celsius and just leave them in there indefinitely. And then when we thaw them out, some of them are going to be still able to be viable. Other methods, primarily here, that are going to keep water away from organisms. So organisms need water for metabolism. So if you deprive them of that water, desiccate them, then they're not going to be able to metabolize. So desiccation, things like, you know, preserving food, like drying out apricots, or drying apples is going to preserve food for a long period of time. Same idea here with lyophilization. This is just a process where you freeze whatever cells sometimes, or maybe food, freeze it really quickly. Usually we would do that by putting it into liquid nitrogen, and then remove the frozen water by a vacuum. So basically this is sublimation. The water goes from being solid straight to a gas. It skips that liquid state. And we can either store cells or whatever it is, like food, indefinitely this way. So this looks like, you know, astronaut ice cream. Basically it's ice cream that's had, you know, all the water removed from it. Backpacking food, or food for backpackers, you can buy dried down, so it's really lightweight to carry. Then you just add some water to eat it. It's supposed to be pretty tasty. But again, you can store things for long periods of time because the microbes need water in order to reproduce. If you've got some liquid that you need to sterilize, then you might want to filter all the microbes out. So maybe you've got something like a drug or a vaccine that you can't heat because that would deactivate it. And so you run it through some kind of filter. So a membrane filter here. You can get membrane filters that have super, super small pores that even viruses can't pass through. And then you have to force the liquid through with a vacuum pump, kind of suck it through. And then all the microbes are going to be caught on that filter, and you're essentially going to have, you know, whatever your drug, whatever is going to be sterile. So there was a case, probably 6 or 7 years ago, of a drug company in New England called the New England Compounding Pharmacy, who was selling, I think it was prednisone, and they didn't have good quality control, and they weren't sterilizing it properly. And so this prednisone, this was a drug that people were, with back pain, were getting the drug injected right into their spinal column. And it was found to be contaminated with a fungus. So people were getting fungal meningitis from this drug that wasn't properly sterilized. And there was like 60 or 70 people that died from this. So it's really important not only to do this correctly, but make sure that you've got good quality control to figure out if you haven't done it correctly. And then, you know, I told you would see this osmosis concept, you know, quite a bit this semester. We use osmotic pressure to preserve foods very commonly. So adding large amounts of sugar or salt to something, yeah, it might make it taste better, but it also provides a really hypertonic environment. So cells that would, you know, spoil those foods are not going to be able to survive because they are losing water from their cells. Okay. Fungus can survive hypertonic mediums a little bit better than bacteria. So it's usually the molds that grow on your food eventually before something like salmonella would. All right, and the last of our physical methods of control, radiation. All right, so we could define radiation as kind of a transmission or emission of energy in the form of light waves or particles through a medium. And there's two kinds of radiation that I want to talk about. And we talked about this briefly already because we talked about using radiation as a mutagen. There's ionizing radiation and non-ionizing. All right, so remember, we get increasing energy the shorter wavelengths we go. Our ionizing radiation here is our X-rays and gamma rays, and maybe even some electrons would be considered to, even though they're particles, they kind of move like waves, so they're considered to cause ionizing radiation as well. And then we'll talk about what happens with the UV wavelength there when we use that as an antimicrobial. But ionizing radiation, what effect does it have on cells? It's going to pull electrons off of other molecules. So, you know, we talked about how, oh, this is a bad thing, and what can cells do to fix the effects of mutagenesis by ionizing radiation? Ionizing radiation also causes other damage to the cell besides mutation. We talked about how it can break the DNA. It can break covalent bonds, and there's just lots of covalent bonds, not only in DNA, but proteins in a cell. It could actually create hydroxyl radicals, which we talked about, a toxic form of oxygen that's going to pull electrons off of molecules. So it just does a lot of different kinds of damage to a cell. It doesn't give off heat. So if you have something that can't be heated, then, you know, hit it with some ionizing radiation, and that might be a good alternative. Depending on what kind of ionizing radiation you use, which wavelength, it can be fast, and it's definitely faster acting than using non-ionizing or ultraviolet light. And it does penetrate better than using the non-ionizing radiation. So we'll look at some applications here of using X-rays, gamma rays, and even electron beams. So let's see, electron beams, or I guess we could just, yeah, electron beams. So a few applications of using electron beams versus, we'll say, we'll go ahead and write this in, gamma rays or X-rays. So remember, the electron beams have the shortest wavelength, but actually the electron beams, they actually don't penetrate very well. And that's sort of the opposite of what you would think. You would think if they have a really short wavelength that they might penetrate easier. But actually electron beams don't penetrate very well. And X-rays, which are a little bit longer, they do penetrate really well, but there's some other, it does take a long time to penetrate. So we don't really use X-rays for anything in terms of trying to reduce microbial numbers. Obviously, in medical applications, it does have a lot of applications, but you know, it's X-rays are going to be potentially causing harm to us, so we have to be really careful the way we use it. Back to electron beams, though. Where do they come from? We have cathode ray machines that are going to be able to produce electron beams, and they actually have quite a lot of applications for things, especially that you don't want to heat, like some kind of like spices, for instance. A lot of application with trying to sterilize medical supplies. So it's you know, you have to, I don't really actually know how long it takes, but you have to, you know, expose them for a certain amount of time. And again, it doesn't require heat, so it's kind of nice that way. Probably the gamma rays have a lot of potential to be used more than what they are already. Gamma rays are emitted by radioactive elements. So things like cobalt 15, I think, cobalt 60 is a radioactive element. So that's the thing that scares people, though. So gamma rays come from radioactive elements, but when something is hit with a gamma ray, it doesn't mean that it makes that thing radioactive, which people kind of associate that in their head. So gamma rays actually penetrate really well. So they can penetrate into solids. It takes a little while for that to happen, but it penetrates really good. Really, again, potential here, not only to sterilize medical supplies, but to shine gamma rays onto food to get them to have a longer shelf life. So this is a really striking example here. This picture was in your book of some strawberries where one group was hit with some gamma rays and one group wasn't. And then 2 weeks later, this is what those strawberries look like. So the ones that weren't irradiated, they're rotting. And the ones that were irradiated, you know, they didn't have any microbes on them. So, you know, they stayed edible for way longer. So it's a really potentially great way to, you know, I'm sure grocery stores have to throw out an awful lot of food because it goes bad, especially fresh food, especially food that we eat without cooking. So lettuce, right? We can't heat it to get rid of microbes. It's a great potential to hit them with gamma rays. Food that has been irradiated is supposed to have this symbol somewhere on the label. The list of foods that are okay to be irradiated years ago was really small. We keep adding to that list as I think the year that we had the big outbreak of, I think it was E. coli in spinach. I think then, okay, now it's okay to irradiate spinach once we had that big, you know, outbreak. So anyway, it has the potential, and it does, some foods it alters the flavor of a little bit. So that's definitely a drawback. And it does decrease, I think it's one of the B vitamins just a little bit. So there are some drawbacks, but if it cuts down on foodborne illness and increases the shelf life, those are pretty good things. Like I said, it's slow to be accepted. people of my generation that hear the term gamma irradiation, this is where we go with that. Like, we think of poor Bruce Banner that was hit with a ray of gamma radiation, or explosion of gamma radiation. And now he's the Hulk, right? But like I said, test after test has proved that food is safe, even when it's been hit with gamma radiation. So, all right, so the other form of radiation, non-ionizing, okay, that's UV light. So that's in that wavelength between about 10 and 300 nanometers. Again, we talked about this before, what kind of mutagenesis this causes, a very specific kind of mutation, pyrimidine dimers, or more often it's the thymines that will bind on the same strand of DNA that will bind to each other rather than their complementary base pair on the other strand. You talked about using UV light this week in lab as well, so should be kind of a familiar topic. UV light doesn't penetrate very well, so it doesn't penetrate through solids at all. Through liquids a little bit, through the air really well, but you have to be kind of close enough to the lamp for that to work. So one great application of this is using UV lamps in water treatment facilities. The UV does a better job of killing some of, especially cryptosporidium cysts. So cryptosporidium is a protozoa that causes diarrheal disease. It does a really good job of killing that where chlorine doesn't. So some of these splash pads that seem to be cropping up, places where, you know, kids can run around and spray each other, that water has to go through an underground UV system to clean that water. It does a better job actually than chlorine of killing some of those protozoan water contaminants. So that's one application. Those lamps are right down in the water pipes, so you have to control the flow kind of very carefully, but it actually does a pretty good job as that kind of final stage in water treatment before, or sewage treatment before it's kind of released back out into the environment. Now UV light is harmful to humans as well. So especially if we look right into the bulb, that's going to be bad for our eyes. It can cause sunburn, it can cause skin cancer. So, you know, we can't be exposed to it directly, but it has its applications. I think in chem lab, maybe they're still shining a UV light on the goggles to kind of clean them off in between people using them. I've seen toothbrush contraptions that will shine a little UV light on your toothbrush. You know, to me, that's like more clean than what you really need. But those sorts of applications are out there. I showed this to my labs, but in hospital settings, hey, this is one more tool to use. You've got a patient that has an antibiotic resistant infection, or maybe an infection caused by a spore former like C. diff, and it's hard to get chemicals every which way to clean. Wheel the lamp in the room, you know, leave the room, turn it on remotely for maybe 15 minutes. And you know, you give it a long enough exposure and it's going to kill any microbe that it comes in contact with. Okay, so turn it off remotely, maybe move it to another point in the room. And again, it's just kind of one more tool to use when things really have to be a certain level of clean. Okay, so those were our, what we would consider physical things that we use to cut down on microbial growth. So now we're going to talk about some chemical control. All right, so even before COVID, oh, we have a question. Okay, so the question is, like, the UV light, when it kills the bacteria, is the bacteria still there? Yes, but if it's not alive, then it can't reproduce. So, you know, eventually it's going to you know, be eaten by dust mites or something like that. You know, it's right. Yeah. Okay, so right, the last unit we talked about transformation, where a dead cell could take up the DNA, sorry, a live cell could take up the DNA from a dead cell. So I guess what you're getting at is, if we're killing these organisms, then is there potential that they can still spread their DNA to other organisms? Yeah, I mean, potentially like in a test tube or potentially in an organism. But eventually, you know, in our bodies, like our immune system is going to eat up all those dead cells and get rid of them. And out in the environment, I don't think that DNA would be able to pass unless it had a medium to pass through. So yeah, like that you're thinking about that and trying to draw connections. Yeah, so did I sort of answer your question? Yeah, so, okay. Okay, good. All right, so even before COVID, our society is sort of maybe hyper interested in chemical disinfectants, and I would say probably using them like way more than what we need to. It's sort of a little bit more understandable when we're in the midst of a pandemic, but oftentimes we are trying to, you know, kill organisms like way beyond what we really need to. For instance, it's really hard to find soap anymore that doesn't have antibacterial chemicals in it. And really, you know, just soap, that's enough to kill a lot of things that we would need to be worried about. So I think we were kind of seeing some of those antibacterial chemicals were starting to be outlawed because some of them were kind of showing up in people's urine and in the environment, places where they didn't belong. So we were kind of starting to understand that maybe we didn't really need all of those, but again, now that we're in a pandemic, it makes it a little bit murkier. So keep in mind, as we talk about our different chemicals, what makes a good chemical? Ideally, you want one that doesn't have very many side effects, that's not going to ruin the surface you're putting it on. Ideally, that we could use at really low concentration, because then it's going to be cheaper. Okay, and ideally, it's going to kill a lot of different kinds of microbes, not just maybe bacteria, but viruses and, you know, protozoa as well. Okay, so pay attention. In these slides, I'm throwing a lot of information at you. Pay attention to how it works. So in other words, does it target proteins or membranes or, you know, everything? Pay attention particularly to uses that we have, and are there any drawbacks to using that particular chemical? Okay, so I'm kind of just throwing some information at you, but pay particularly attention to those things. Okay, so I have these organized by low level, medium level, and high level. So a low level antiseptic or disinfectant is going to work against those microbes that are easiest to kill. And a high level one will even be effective against spores. Okay, so that's why I've got that organized that way. None of them are perfect. They'll have some disadvantages. Okay, soaps and detergents seems like a good place to start. That's probably the antimicrobial that you use the most often. Incidentally, soaps and detergents aren't exactly the same thing. Soaps are made from a natural product, and detergents are always going to be kind of synthesized. So there is a difference between those two things, but they both work the same way. They're surfactants. So that means that they reduce the surface tension of liquids. Okay, so kind of keep molecules from being attracted to each other. So they're good at decreasing the attraction between molecules. That's what I'm trying to say. They are particularly good at disrupting cell membranes and walls. Right? So something, again, like a soap, that's a surfactant. If it's breaking apart, attraction between molecules, we're soaping up our hands and then we're rinsing it away. Okay, that helps. That mechanical action just helps. Again, in the water, rinsing it away, it helps pull apart any microbes that might be on our skin, as well as dirt. Something like a detergent. Here's an example of a detergent, quaternary ammonias. We often refer to them just as quats. What are quats? Basically, an ammonia ion here has 4 hydrogens like this. Okay, so a quaternary ammonia actually replaces these hydrogens with some other functional group. So I went back through. In the future, your examples in your book are a little bit better than this one. This one's kind of hard to see, but there's actually here, there is a valence of 4 here. And the 1 hydrogen is going to be replaced with this huge, hydrophobic group. So this long arm here is going to be really good at inserting into membranes. Okay. This is a low-level disinfectant or antiseptic, depending on how it's used. So it's not going to affect endospores or mycobacteria. And kind of alarmingly enough, this has been quaternary ammonias have been used for so long that we're starting to see some resistance to quats, especially with a particular bacteria, Pseudomonas. That's a bacteria that we use in lab all the time, and it's typically not one that will cause infections in healthy individuals, but it's a huge problem in healthcare settings. Burn victims have a huge problem getting Pseudomonas infections. It's an organism that just has a lot of natural resistance, not only to antibiotics, but again, to something like a detergent. That's kind of a problem if we're starting to see organisms become resistant to the disinfectant that we're using. I don't think that that's widespread yet. I think that's just something that we have noted at some points. Okay, so that's our only low-level chemical that I'm going to talk about. There are some other ones in your book, like the use of heavy metals and phenols. Phenol used to be considered a gold standard. Like that was the disinfectant that everybody used for everything. It was the one that Lister would spray in the operating room to cut down infections in the operating room. But that phenol, when he sprayed it in the air and had doctors wash their hands in phenol, it made doctors' fingernails fall out. It turned their hair white. They had respiratory problems. So it's one of the reasons that we don't use phenol very much anymore, is because it's so irritating to us. So we found other things in a lot of cases that are better than phenols, and heavy metals, the same thing. They're just so irritating to us that their use is really quite limited. So I decided to skip over those and move right to the halogens. These are considered an intermediate level antimicrobial. So here's an example of the halogens, right? These are elements that are off to the kind of right side of the periodic table. Their outer shell is almost full. So they want to fill them up. Okay, so they are grabbing electrons from other molecules in order to fill their outer shell. Okay, so they're fairly reactive. They're strong oxidizers, in other words. They're grabbing electrons from other molecules. All right, so chlorine, iodine, bromine, and fluorine I'm going to talk mostly about chlorine and iodine, but bromine, some uses of that. A lot of swimming pool, hot tub owners are using bromine instead of chlorine now, just because it evaporates a little bit slower. so it works a little bit longer, maybe a little bit less irritating to skin and hair. Fluorine, obviously we add that in really tiny small amounts to our drinking water. It's in your toothpaste, really small amounts, and that helps cut down on tooth decay. Okay, but in general, all these halogens, oh, I guess I didn't need to write it. It was right there. They're strong oxidizers. They're also breaking disulfide bonds within proteins. So they're acting especially to damage proteins. And I was looking at this before class. I didn't really like the way that I wrote this. Kills on surfaces within 30 minutes. I'd say if it took 30 minutes to kill, then that's not really a very big advantage. More often, they're going to kill much more quickly than that. But it does take several hours for the halogens to kill endospores. So that's why they're still considered medium or intermediate level antimicrobial rather than high level. So they do kill spores, but not within sort of a reasonable amount of time. Okay. All right, so let's look at chlorine first of all. Really commonly used antimicrobial. Usually we use this as a disinfectant. That might not always be the case. Obviously, chlorine in swimming pools, it gets on your skin. I have heard of people with recurring skin infections, doctors prescribing them to sit in a bath with a little bit of bleach in it. But commonly, we're going to use this, again, on non-living objects. You know, when we use it in a pool, we're using it in the water to cleanse the water. And it is quite susceptible to organic loads. So you got a big mess to clean up. You got to clean up the mess first and then bleach it to make the disinfection work. One disadvantage is it's quite sensitive to light and pH. So when you buy bleach, and by the way, hypochlorite, that's kind of the technical term for bleach. When you buy bleach, you're always going to buy it in these opaque containers. That's the reason is that light will make the bleach basically dissociate into chemicals that aren't going to have any antimicrobial power at all. So we buy it, and that opaque container slows down that dissociation, but really, after about 9 months or a year, that bleach has gone off. So if you buy bleach, buy it in a really small container, and then buy more when you're out of it. So to stock up and buy a whole bunch, it will probably go bad before you can use it, unless you're using it on a regular basis. So if you're using some bleach to try to get rid of stains on your shirts and it's not working anymore, it's just because it's old. Okay, so it does go off. Like I mentioned before, if the pH isn't right, it dissociates pretty quickly as well. We use bleach in lots of different forms. The elemental chlorine is most often used at water treatment plants. Chloramine is chlorine and ammonia liquids. So maybe that raises a red flag. When I first read that, I thought, oh, that's crazy, because I know that you can't use bleach and ammonia. You should never mix those two things, because that makes a toxic gas that can actually kill you if you do that enough. So, I mean, you can buy ammonia in the store. It's kind of easy to miss. There might be like one brand of ammonia. It's always best to never mix cleaners. just use one cleaner and don't try to mix them, because again, that can be a potential fatal combination. But when they're mixed as a liquid, like in the company, this actually makes the chlorine a little bit longer acting. Okay, so in other words, the killing power of the disinfectant keeps going, lasting for quite a long time. It's slower to kill, but it does last a little bit longer. So that might, have a little different applications. Of course, bleach. This is what we're going to use most often. Around the house. We used to use bleach in the labs to wipe the bench down. It is very fast acting. It's effective against a lot of things. When you want to, you can actually use bleach as a gas if you want to disinfect some kind of large space as well. Oh, let's see. What did I forget to say about that? I feel like something, but okay. The other halogen that has quite a lot of uses, iodine. Okay, and again, we tested some of these in lab this week. It has the advantage that it's a little bit less susceptible to being deactivated when it's in with some blood or vomit or whatever. Common uses of iodine, and there's different forms again that we use this in. Often used as an antiseptic in wound treatment, or maybe pre-surgery or pre-needle poke. Campers can use iodine tablets to treat water. So obviously it's very heavy to carry water around with you, or maybe even a filter is heavier than what you want to carry. You can't use iodine tablets. Now you have to get it in the right concentration in your water. You got to measure it carefully. And then you have to let it sit for a while. But if you get it in the right concentration, then you can drink it and it doesn't taste too bad. The big drawback with using that is that at the concentration that you use it, and it's okay for us to drink, it's not high enough to kill all the things that might be in that water. So there's some protozoa that are going to be able to withstand that iodine that could still make you sick. So it's not really the best way to treat your water, but like if you don't have any other options, it might be helpful. Some disadvantages with using iodine. It's a little bit irritating to our skin depending on which form you use it in. Obviously it stains. We had a little spill in lab this week. So if you walk into room 303, you're gonna see where that iodine spilled. That'll be there for a while. And some minor allergies associated, with using it on our skin. Okay, so we can use iodine in a couple different forms. When it's used as a tincture, that means it's dissolved in alcohol. So tincture of iodine, you may have heard that before. We can dissolve iodine in water, but apparently it doesn't work quite as well when we do it that way. So we often find it dissolved in alcohol. We can also use iodine in what we call an iodophore. And that means the iodine has been attached to a carrier protein, and that makes it last a little bit longer, as well as being less irritating to our skin. So very often, iodine used in a medical setting is used in the form of betadine. That's kind of the brand name. Again, that's iodine that's linked to a carrier protein. And here you see that being used on a hand before hand surgery. Might use that before a needle poke. Again, just a little bit less irritating. The other applications beside medical application is in dairy. So I have some family members that have a big dairy herd. And I asked them once, well, how do you clean off, the udder before you do milking? And they said, oh, we use something called teat dip. And that sounds a little, vulgar or whatever, but that's what they call it, teat dip. They go down, they hose off the cows, and then they go down and they kind of squeeze that bottle so that the iodine gets up on the teats. And then they go down the row and do that to all the cows, or 5 or 6 of them or whatever. And then they come back and they wipe it off. So it gets a little bit of contact time. Again, just one more way that we try to make milking as hygienic as possible. that picture, that's like the cleanest cow I think I have ever seen. But again, just one more way that milking is pretty hygienic. All right, so we just have a couple chemicals to finish up for next time, and then we're going to get into our antibiotics. I hope you all have a good weekend, and thanks for listening.